We're going to continue talking about cells, and you're probably like, when are we going to get to talk about human stuff? Well, we have cells, we have water, and we talked about the importance of water in our cellular systems. But for all of our cells to be alive, we have to have water, we have to have nutrients. I mean, that's not surprising, right? We have to produce energy. And we need oxygen. But on the flip side of this, and much like we talk about biology, for everything you have, there's typically going to be an opposite. And so while we need these, we also have to remove the extra water we don't need, because we are converting our energy, our raw materials into energy. We're going to have to get rid of CO2. That's a breakdown product of producing energy. And we also have nasty byproducts of that metabolic process of recovering the energy from the food we eat. So we have to get rid of those wastes. So to get material into the cell that we need and out of the cell that we don't need, we have to do transport. And we have to transport materials across the plasma membrane. You remember our plasma membrane we talked about last time? If we had to look at the molecule that is most abundant in the plasma membrane, what category of molecule is most abundant in your cell membranes? We had proteins, we had nucleic acids, we had lipids, and we have carbs. What's the most abundant of those four? We'll start there. Lipids. Lipids are the most abundant. Now we had three categories of lipids: triglycerides, phospholipids, and steroids. Which of those is the most abundant in the plasma membrane? Phospholipids. So because of those phospholipids, and you remember it's a bilayer or Oreo cookie? Because of that, we're going to have special molecules and proteins that can help getting certain molecules in. Some molecules are going to be able to get in on their own, and we're going to talk about those. But as we look about this phospholipid bilayer that is our plasma membrane, we say that membrane is selectively permeable. That's a big fancy way of simply saying some things can go in and out on their own, but some things can't. And so when we look at oxygen and carbon dioxide, those molecules are going to be able to freely move in and out of the plasma membrane with no one else or nothing else helping them. So that means we're going to have a selective transport of other materials. Those that can't move freely on their own. And those that can get in pretty freely are ions, organic molecules that are nonpolar. But some macromolecules, most of these macromolecules, and you know that word, macromolecules, just means big molecules, right? So things are too big, they're going to have help getting into and out of the cell. So all of these things are about transport, whether it happens on its own or whether it has to have help getting in with the assistance of a protein. This is all characterized as transport into and out of the cell. Now we're going to break down transport into two major categories: passive transport and active transport. You know, like I said, I like to start simple and build on that. What do you think the real major difference is between passive transport and active transport? If you're passively sitting there, are you using a whole lot of energy? No. But I rode a bike at the HPE for 10 miles. There's nothing passive about that. I use a lot of energy. And so the difference, passive transport, you don't have to use energy to move that stuff. Active transport must use energy. Those are the two big differences. I mean, that's the big difference. Now, we're going to see other differences, sure, but that's the big one I want you to home in on right now. Now, when we get to passive transport, we're going to talk about diffusion We're going to have simple and facilitated, and then osmosis, which is a big fancy word for simply the diffusion of water. And then with active transport, we're going to have direct or indirect. I like to call these primary and secondary. Those are terms that we use more often now. So this is sort of the big global picture, and now we're going to move into each one in the specifics. So first, passive transport, diffusion, and remember I said osmosis was simply the diffusion of water. Since it's passive, what do we not have to have? Energy. And with passive transport, not having to use energy, I like to compare this to riding a bicycle downhill. Do you need to use a lot of energy if you start at the top of a hill and you're just riding that bike downhill? No. And so in much the same way, our molecules are going downhill, and that hill is their concentration gradient. Does everyone understand what I mean with concentration gradient? At the top of the hill, you got a lot of this certain molecule. At the bottom of the hill, you don't have any. Or in instances of our plasma membrane, you'll have a lot of the molecule on one side of the membrane and none on the other. You see the gradient? High to low, just like riding a bicycle downhill. And so in the case of diffusion, this is going to be the movement of solutes. Have you ever heard that word before? That word's a big fancy science scary word for stuff. Stuff other than water. Solutes are what you dissolve in water. So one solute, we could think of salt. Another solute, since I had unsweetened tea yesterday, and I don't even know why they sell that stuff, you put sugar in that business. So sugar would be a solute. So in this case, if we look at our example, here we have a selectively permeable membrane. And in this case, that membrane is going to be permeable to our solute. It's not permeable to water. Do you see how over here inside A we have a high concentration of our solute, the black dots? We don't have as many on the right side in B. If you let that sit over time, those molecules are going to move, diffuse through that membrane. And what can you say about the solute concentration on either side now? They equalized. They reached an equilibrium where you have just as many on one side as the other. They have diluted themselves out equally on each side of that selectively permeable membrane. We had a concentration gradient here, high to low. When we don't have a gradient, when it's equal, the net movement of those solutes is zero because they're at equilibrium. They've reached a balance. We say that term a lot about the human system, and now in cells, they want to be in balance. Now, osmosis is a little different because with osmosis, we're talking about the diffusion of water. Water is going to move toward the highest concentration of solutes. And so in this case, let's look at A and B again. We have a high concentration of solutes on A, low concentration in B. Our membrane now is not permeable to the solutes. And we're going to use salt in this case. Water can move across the membrane, but the solutes can't. So what happens if we let that sit over time? The water molecules move in the direction of the greater concentration of solutes in an attempt to do the same thing that happened here. They're trying to dilute out the solutes equally on each side of that membrane. And if you've ever been in salt water or played around with salt water, you know it can dehydrate you in a hurry. Because if this is salt water and this is you swimming in the water, and this membrane is your skin, what's happening while you're swimming in that salt water? You're getting dehydrated. And that's why as soon as you get out of swimming in the ocean, you're thirsty. Yeah, even more so than swimming in the lake or swimming in a swimming pool. So that's the movement of stuff. That's the movement of water. Now let's apply this to our cells. And our selectively permeable membrane that's going to allow oxygen to freely move, CO2 to freely move, water to freely move. And so all of these molecules that can cross the plasma membrane without the use of energy, downhill, following that concentration gradient, they're moving by diffusion, or in the case of water, osmosis. And again, so typically when we look at those molecules, they're going to be small, non-polar molecules. Water, slightly polar, it can still diffuse. And in some cases, we're going to have special pathways so water can diffuse even more rapidly. So if you cross the plasma membrane unaided, that is said to be simple diffusion. I mean, if you were to leave off simple and say diffusion, that kind of means the same thing. It's like the one before the variable in algebra. It's understood. But if we're going to talk about this next one, you have to put a word in front of it, because now we're doing something different. Let's look at diffusion first, that word. Are we having to use energy? Do we use energy if molecules are diffusing? No, I see a bunch of shake. No, you're right. So it's passive transport down its concentration gradient. When we put facilitated in front of diffusion, all this simply means is that the molecule can't by itself cross the lipid bilayer and diffuse in and out of the cell. It has to go through a protein. So you can think of some of these proteins like drinking straws that go through the membrane. That drinking straw creates a tunnel that's going to allow larger molecules, maybe polar molecules, to pass through that lipid layer in this channel that allows it to. And some of these channels are going to be very specific about which molecules can pass through that opening. Now, since I mentioned the drinking straw, that kind of transport protein is called a channel protein. And again, since we're talking about polar molecules passing through, it's not surprising that the middle of that channel is going to be hydrophilic. What can you tell me about the middle layer of our membrane? That fatty acid area? What can you tell me about that area? Is it hydrophilic? You know, our fatty acid chains, they're a bunch of hydrocarbons. It's hydrophobic. So you see, we've changed the characteristics of the passageway here for our channel to allow molecules that are polar, that are hydrophilic, to pass through that could not undergo simple diffusion. Now, in some cases, we have proteins that are called carrier proteins. This is where it gets tricky, so listen very carefully. Because you will have proteins bind, or transport molecules bind to these proteins. And the proteins act like revolving doors. As soon as that transported molecule binds, that protein changes shape. So that the protein can, let's say that's the outside of the cell and that's the inside of the cell. This carrier protein can grab the package, ion, carbohydrate, whatever. And that protein can change shape so that it moves that transport molecule to the inside of the cell. So now we have a protein moving. This sounds like that protein's using energy, doesn't it? But it's not. That protein is not using any energy. And the direction of transport, when we get this package and we move it in this direction, which direction is our concentration gradient? Since we're still talking about diffusion, high to low, it's still downhill. Even though we have this facilitated term here, even though we're talking about a protein that changes shape, it still doesn't require energy, and it's still moving the molecule down its concentration gradient. You see the difference between simple and facilitated? Simple doesn't need help. Facilitated has to have a protein to help it. And that's the only difference. Now, when we look at our proteins, and whether we're talking about channels, like our drinking straws, or carriers that change shape, I mentioned they were pretty selective in what they will move and what they will transport. So we can define these proteins also by the number and the direction with which these molecules are transported. So if we transport one molecule in one direction, we say that transporter is a uniport transporter. How many wheels does a unicycle have? One. So one molecule moves in one direction. Now, some carriers and channels can move two molecules, but they move them in the same direction. And that type of transport is called symport, because sim means the same. And then contrary to this, we can move two molecules in opposite direction, one coming in, one going out. And that is an antiport mechanism, moving two, but in the opposite directions. So, we've characterized so far simple diffusion, facilitated diffusion, which is either a channel or a carrier protein that has to be used. And we know those are forms of passive transport. Now we can add a classification based on the number and direction with which we move those molecules. See, we start simple with active and passive, but we're getting more complicated as we go. Does everyone see what we've done so far? So here are some examples of our facilitated diffusion. What kind of transporter would you say this is? What kind of transporter would you say? Or what kind of transporter are we looking at? It's kind of a trick question. Huh? Sorry? Symport. Now we're thinking symport same direction. How many different molecules are we transporting? They're all those cubes. So that's one molecule going in one direction. What kind of transport is that? That would be uniport. What if I told you that was an incorrect answer? I was looking for facilitated diffusion. How would you have known what I was looking for? Have you ever taken a test and you read the question on the test and you go, there could be 3 or 4 correct answers for this one. And you get your paper back and it's wrong because it wasn't the one the professor had in his head? It's going to be important that you read the questions on my test. Now, I purposefully left that question vague. But if I were to say, based on the number of different molecules and the direction of transport, how would you classify that? What would you answer then? Uniport. But if I say, in this illustration, what type of transport is happening that's utilizing the protein? You see the focus. The focus is going to be on the question. All right, that was the big passive transport. Now we get to active. Passive, no energy. Active, we're going to use energy. This is going uphill. My least favorite way to ride a bike. But with that analogy, though, too, now diffusion went downhill, high to low. Guess where we're going with active? Low to high. We're moving molecules against their concentration gradient. That's why we can't use diffusion, because they're going the wrong way. Now, since we're going the wrong way and we can't undergo simple diffusion, we have to use proteins also. But do not confuse our facilitated transport use of proteins with active transport use of proteins. Okay? Now, here are 5 steps. These are general steps for active transport. And notice, I've already stuck primary in here. We'll get to the difference in just a second. But I want you to follow these steps. The first thing that's going to happen is that molecule to be transported is going to bind to the protein. It's not going to be a channel in this case. It's going to have to change shape. So we don't have any active transport channels. They're all going to be carriers. So the first thing, you bind to the carrier. that carrier protein is going to have to capture some of this ATP, adenosine triphosphate. When we're talking about cells and we're talking about the human body, when you hear ATP, what word should you be thinking? Energy. Energy. The energy that all of your cells in your entire body use is stored between the bond of that third phosphate and the second phosphate that stays attached. This is adenosine diphosphate, that's adenosine triphosphate. The way cells recover the energy is they clip that third phosphate off. The energy that was in that covalent bond between those phosphates is released and the cells can use it. So we bind to the molecule in A. We release the energy in that bond. That energy then is what is used to change the shape of the protein. Conformational change, a big fancy word for just changing the shape. Now, since that protein that's changing shape already bound to that transport molecule, that changing of the shape is going to move that transport molecule, that package, from one compartment to the next compartment. And then even though I've got this put together in D, I want you to make number E or number 5, this last sentence. The carrier resets. Bind the molecule, release energy, change shape, move the molecule, reset. Here's our example. Binding, there comes our energy, changing shape, releasing, resetting. Binding, energy, change shape, release, reset. There's only one thing I don't like about that illustration. And when you're looking in your textbooks and looking on Google and Wikipedia and stuff, be very critical how you think about these illustrations. What's wrong with that example of primary active transport? Look at our concentration gradient. It's going the wrong way. High concentration out here, low in here. That's wrong. For active transport, it's got to be opposite, right? So be careful. Hey, I like the animation of this part, so we'll pretend there's a lot of red dots in there already. Or take your marker and put some more, okay? Primary active transport. Now, this is where it gets a little bit trickier. We're going to build on that. Because in practically every cell in your body, you have this pump that's called the sodium-potassium pump. You would not be here without the sodium-potassium pump. In fact, the energy used by the sodium-potassium pump in your body accounts for 12% of your energy use every day. That's a lot. That's a lot of energy used just for all of these molecules in your muscles, in your heart, in your kidneys, in your nervous system, everywhere. And so when we look at primary active transport, remember we're going to bind those transport molecules. So we're going to bind 3 sodium ions. We're going to bind 3 sodium ions that are inside the cell, and we are going to move them out. In active transport, am I going to have to use more energy to reset? No, just that one molecule. But here's the thing with the sodium-potassium pump. This protein gets stuck in this position. And there's only 2 things that's going to unstick me so I can reset. And those 2 things that unstick me are 2 potassium ions outside the cell that I need to move back. I've released the sodium. Now I'm going to pick up these 2 potassium. That 2 potassium are the key to unlock me in the stuck position so I can reset. And when I do, I drop those 2 potassium inside so I can do that whole thing again. The sodium-potassium pumps change in shape anyway. We're not going to use 2 separate proteins then to move the sodium out and the potassium in. We're doing that anyway. But you see why we distinguish primary and secondary? The moving of the 3-sodium would never happen if we hadn't used that molecule of ATP. But for our potassium, you were going back anyway. We didn't have to use any more energy, so that's secondary active transport. Now, we can also define this pump because two different solutes going in opposite direction. What can we call that? Antiport. And there it is. Again, ignore the concentration gradients. They didn't do it right. Use our energy, change shape, release, but we're stuck. There's our 2 keys. Unstick us, release them in, and we can do it again. Primary active transport. Secondary active transport because we didn't have to use any more energy. You see the difference? All right. That's sodium-potassium pump. Now, the last couple of things to cover here is sometimes cells will gulp stuff. You ever seen those videos of the amoeba that's kind of creeping and crawling around and it comes up to a bacteria and it just engulfs it? That's like cellular eating. And cells in your body do kind of the same thing by a process that's called endocytosis, especially your white blood cells. Many of your white blood cells are like amoeba. They can crawl around, and when they find the bad stuff, they will engulf it, forming this little vesicle, this little bubble, until it's completely inside the cell and trapped. So that's endo, bringing in, cyto meaning cell. But then cells can also do the reverse. Cells, especially your glands, secrete a lot of stuff. And they basically do this process in reverse, and that's called exo or exiting. Here we have, we'll say this is a bunch of hormones we've got to release. It's in a little vesicle. Well, that gets closer and closer to the membrane until it fuses with your plasma membrane, and all that stuff is released and secreted outside the cell. So again, if you learn one picture, they're just the opposite. Look, the only difference are the arrows going in different directions. Endo, bringing stuff in. Exo, taking stuff out.
Is an example of that like us sweating?
You know, sweating is an example because we have sweat glands, and those sweat glands have cells that will have really hydrated, salty solutions, and that material in those cells get pushed to the surface. This would be the, let's say it's the inside of our sweat duct that comes up to the surface of our skin and spews all that stuff out. Yeah, exactly. That's sweating. I sometimes call that cell spitting too, because the cell's getting rid of spitting stuff out. This is a cell drinking. All right. So, diffusion, passive transport, active transport, primary and secondary, and our endo and exocytosis. Those are our principal means with which we're going to transport materials. So you need to be familiar with the differences and the similarities between all these different transport methods. All right, now we're going to get into our organelles. We've talked about plasma membrane. Now we're going to cover the others. And not necessarily all of these, because some of these are going to be plant cell related. But we're going to take each one of these, and we're going to take kind of a big chunk of them in a group that we put into kind of like an organelle system, if you will. So we're going to cover each one of these as we go down, with the exception of vacuoles and chloroplasts. That's plant stuff, and this is human biology. Plants are for salads, right? I'm kidding. Don't tell the botanist I said that. So, we're going to take that list and we're going to group some together first into what we call the endomembrane system. Now, this is a slide that I've used from another class, so I want to explain eukaryotic. Have you ever heard that term before? What does that mean? Eukaryotic cell. Has a nucleus, a membrane-bound nucleus. What do you call cells that don't have a membrane-bound nucleus? Prokaryotic cells. That's our bacteria. Talked about that a little bit when we had our domains and classification. Well, with our endomembrane system, we're going to see that that includes the nucleus, our endoplasmic reticulum, and actually these two things are physically connected to each other, and it's kind of cool how that happens. Then even though they're physically separated, functionally speaking, the Golgi complex is a part of this system. Oops. And then we're going to have a lot of these little membrane-bound vesicles. Some of these just transport materials back and forth, and some are going to be used to digest material that might be brought into the cell. So the nucleus, that's where we have our DNA, right? Nucleus DNA. Endoplasmic reticulum. I want you to remember this can be a location for protein synthesis, making proteins, but it absolutely is the location to make more membrane in the form of lipid. Golgi complex, I think that's pretty straightforward there. With our proteins, we're going to modify, sort, and package our proteins to ship to different places in the cell. And again, we're going to get into more detail than just these are the highlights right here. This is the Cliff's Notes, or what's the short version now? It was Cliff Notes in my day. If you don't want to read the book, what do you still look up Cliff's Notes? Huh? Spark Notes. Yeah, this is Spark Notes. I couldn't remember that one. So this is the Spark Notes version. Now also, I mentioned those little vesicles, the little membrane-bound bubbles. Those can be lysosomes. And again, that's going to be for digesting material, as well as peroxisomes. Can you guess what's in a peroxisome? Peroxide. Yeah. That nasty stuff Grandma used to put on you, bubbles up your cut. Yeah, peroxide. Again, there's some white blood cells that have these superoxide ions that they can actually launch at bacteria and stuff. It's like throwing hand grenades at them. And they're filled basically with hydrogen peroxide. So these vesicles, including the transport vesicles, we haven't listed them there, but including transport vesicles, are part of what we put together in this organelle system that we call the endomembrane system, or essentially the membranes that are inside the cell. So again, that's the Sparks version. Let's look at the more detailed description. So our nucleus, it's where we have our DNA. What you're looking at here is an electron microscope picture. Very, very high magnification. And this line that you see that I'm kind of outlining here, that's the membrane of our nucleus. We usually have this really dark spot on the inside. It's called the nucleolus, but we'll talk about that more in a minute. But what I want you to understand about this nuclear membrane is it is a double membrane. Did you hear what I said there? It is a double membrane. When we talk about our plasma membrane, we usually call it a lipid bilayer, right? Two lipid layers put together. Here, we're talking about two whole membranes. So how many lipid layers surround the nucleus? 4. Do you understand what I'm talking about? These two lipid layers with the outer membrane of the nucleus, these two lipid layers with the inner membrane of our nucleus. I'm glad I was a Trekkie as a kid. I'd never be able to do that. The other thing that I want you to remember about the nuclear membrane is it contains holes. And if you were to look, let's just go back for a second. You see this purple, that's our nuclear membrane. See all the black spots? Those are holes like a wiffle ball. But they're not just opening that anything can move in and out. These pores are very highly regulated. Material has to be transported in and out. It's like these pores have bouncers to check your ID, make sure you're supposed to be coming in and make sure you're supposed to be coming out. Not everything can simply move in and out of the nucleus. Now, with this picture, the reason that it's so much darker on the inside is there's more stuff. It's got a greater density. And the more stuff of our nucleolus is we have a lot of RNA being made. Scratch that out and write site of RNA synthesis. Your ribosome is largely RNA, but I want it to be very clear. This is a lot of RNA that's going to be used to build our ribosomes. And it collects in this dark spot in the center of our nucleus called the nucleolus. Now, in our nucleus, again, that's where we have our genetic material, our DNA. But to use that code that's contained within our DNA to build a protein, the proteins are built in the cytoplasm. DNA can't come through those pores. So the solution that nature has come up with is that a copy of the code is made in the form of RNA, which can be transported through the pore. So in our nucleus, we convert DNA into RNA. The RNA then comes through the pores of our nucleus into the cytoplasm, where that code then can be used to build and assemble the amino acids into protein. I want you to go ahead and get accustomed to these terms. We're not going to see them until chapter 17, but this is test 1 material as well. All right? The process of making a molecule of RNA from DNA is called transcription. The process of making RNA from DNA is called transcription, and it happens in the nucleus. Once the RNA gets into the cytoplasm, that RNA code is used to assemble our amino acids into proteins, and that part of protein synthesis is called translation. Transcription, translation. Don't you love it when two words sound so much alike, but they mean different things, right? We did that just to trick you up. Transcription in the nucleus, making RNA from the DNA. Translation happens in the cytoplasm, and it's taking that code within the RNA, using it to assemble our amino acids into proteins. Transcription, translation. Only differs by a couple of letters, right? So in this process of making proteins, which has to come first? Making the RNA or coming out in the cytoplasm to make our protein? Which comes first? Making the RNA. Transcription. That's got a C in it, right? Transcription. Translation happens in the cytoplasm. That has an L in it. C comes before L in the alphabet. Maybe that could be a way to help you remember which is first. We're going to have a more technical way to talk about it when we get to 17, but I wanted to introduce that here. Maybe that will come back as we talk more and more about that in chapter 17. So here again is another illustration of our nucleus. There you can see our nuclear pores. There's the inner membrane. We have the outer membrane, and that white is simply the inner membrane space. It's the space between our two membranes of our nuclear envelope. Now, this is the beginning on the outside here, these folds. This is the beginning of our endoplasmic reticulum. And I've got this here to illustrate how the lumen in here, the inner membrane space of the nuclear membrane, is continuous with the inside of our endoplasmic reticulum. And so that's another reason why we have included this together in this endomembrane system. Now, this is a biology class, so don't get uncomfortable when I use the E word. Okay? You know what the E word is in biology? Evolution. I know a lot of people get nervous when I talk about evolution. But this is a biology class, so we got to talk about it. When you look at the nucleus, and you look at these two membranes, and even when you look at the distribution of the proteins on the surface, there is a hypothesis that in the distant past there were two separate cells. One cell was really good at making energy. But this other cell was really good at having all of this DNA, this genetic code to keep things straight. And so these cells got together and said, hey man, we need to set up a partnership. Let's work together. And so the cell that could make a lot of energy brought this cell that had all this genetic material, brought it inside of itself to create a symbiotic relationship. So the inner membrane of our nucleus is more representative of the initial membrane of this separate cell and its plasma membrane. The outer membrane of the nucleus is more similar to the plasma membrane of a host cell that would have brought it in. I mean, when you look at all the evidence, it's pretty compelling that, this is a plausible biological hypothesis for why we have this double membrane as part of our nucleus. And evidence is building more and more that that's a plausible way for that to have occurred. Now, I mentioned the nuclear pores. They're not just holes like an actual wiffle ball. They're actually over 100 proteins that regulate the material that can pass in and out of those pores. In fact, you have to have a chaperone that takes you in and out of the cell. The chaperone that has to bind to molecules that are going to be transported is called important. Isn't that kind of creative how they named it? And the protein on the inside that chaperones and transports molecules out is called, guess what? Exportin. And if those molecules aren't present, then these 100 proteins in that pore are not going to permit it to pass in or out. But it's good that RNA is one of those molecules that absolutely gets transported out. All right, now let's move to the next part of our endomembrane system, the endoplasmic reticulum. Now, our endoplasmic reticulum comes in two flavors, rough and smooth. The smooth endoplasmic reticulum is where you make more membranes. It makes more lipids. But most people are more familiar with what is termed the rough endoplasmic reticulum. And what makes that ER rough are these little protein factories called ribosomes. And when ribosomes are present all over the surface of the endoplasmic reticular membrane, it's called rough ER, and you can see this little blue line right here. That is a protein that's being produced. It's almost like a pasta maker. Have you ever made pasta? Okay, have you ever played with Play-Doh? You stick it in there and push the handle and it squirts it out? That's what ribosomes are. They're squirting out protein. And in this case, the protein gets injected into the middle of our endoplasmic reticulum. Not all proteins. Some of these ribosomes are just free-floating inside the cell. But if these ribosomes are on our endoplasmic reticulum, we say it's a rough ER, and the proteins that are being made are either going to be secreted outside the cell, or these are going to be proteins that are present in some sort of membrane, either the cell membrane, nuclear membrane, or a membrane of our other organelles. If you're just going to be a protein free floating around in the cell, you're just going to be on a ribosome floating around somewhere. Now, notice this as well. In the ER, especially the rough ER, glycosylation begins. Glyco. What do you think of when we say glyco? Glycogen, carbohydrates. So proteins have carbohydrates added to them to get to their final form, and you start adding a few rudimentary building carbohydrates to the proteins in the endoplasmic reticulum. Most of the protein modification and the addition of sugars is going to happen in the next step, but it has to begin in the endoplasmic reticulum. So here's a nice illustration showing the rough endoplasmic reticulum with the dots all on it, and see it connected back to the nuclear membrane. And then here we got this, it looks kind of like a pipe organ. This is the smooth endoplasmic reticulum, and this is where we're going to be making a lot of our lipids, and essentially we're making more phospholipids, assembling them together to produce more more membrane. Because as we transport materials in and out of the cell, sometimes we're going to be losing membrane. So we've got to make more of it so the cell stays the same size. And so the generation of lipids, the production of more membrane, that happens in the smooth portion of our endoplasmic reticulum. Now, here's our little ribosome. This is not necessarily considered a membrane-bound organelle. It's kind of an honorary organelle because of its relationship with the endoplasmic reticulum. But what I want you to remember about our ribosomes is that this is basically the warehouse where proteins are made. And our ribosomes consist of two units. It's almost like cupping your hand together. Have you ever done that in a swimming pool? You put your hands together and when you squeeze them, water shoots out? That's because you have a space inside your hands. Well, when you put these two subunits, a large and a small, ignore the 60 and 40. I mean, that's just kind of a size thing. You got a large one and a small one. That space inside the ribosome is where your RNA is going to slide through. That code's going to get read and it's going to be translated, and amino acids are symboled like beads on a string inside the ribosome, and it's going to get pushed out as the finished protein product. Here it's trying to illustrate the protein being extruded. This is going to be your RNA that's sliding through, and that's just the two parts of your ribosome. So protein production, making protein, happens on the ribosome. So endomembrane system, nucleus, connected to the endoplasmic reticulum. Endoplasmic reticulum is where we started making proteins because of the ribosomes that were on the surface. Those membrane or secreted proteins leave the endoplasmic reticulum in little vesicles, little bubbles. It's kind of like exocytosis. All this material gets to the end and it starts to push out on one of the membranes of the ER until a little bubble pops off. And that's a little transport vesicle bubble. And that material in that vesicle moves from the endoplasmic reticulum to this basically pancake stack of membranes. It's called the Golgi apparatus. Golgi was the guy's name that discovered this, so he stuck his name on it. And what you can see in this Golgi stack, we have some of these little bubbles on this side, and we've got some little bubbles on this side. You can see quite a few of them here on the lateral surface. So on one side, these transport vesicles from the ER, as they get closer, they all melt together until they form a layer. And that layer moves from the bottom of the Golgi toward the top. And this material inside these layers of Golgi, these proteins are getting modified. Their sugars are getting added to it. The proteins are getting folded. Other chemical changes are happening. And then as we move from the bottom, the first arriving, as we get closer to the top, that top layer begins to disintegrate into other little bubbles, other transport vesicles that then have instruction to go, I have to go bind to the cell surface to be secreted. I have to go bind to the nuclear membrane. I've got proteins that are going to be important for making new pores. I'm a vesicle. I'm going to stick around because I've got enzymes in me that's going to help digest a pathogen that I've brought in by endocytosis. So that's part of the modification, packaging, and sorting that is going on inside these layers of our Golgi apparatus. These are the layers that are breaking apart and going away. This is the layer that's just recently formed, and we're going from very young layer to the oldest layer as we move through the stack. It's almost like a conveyor belt. You're continually making new layers, and we're continually getting rid of the old ones. Well, what's the big message about Golgi? Processing, packaging, and sorting of our proteins. So this illustration sort of puts it all together. Here's our endoplasmic reticulum in blue. We're going to make that the rough ER. You see how we've got these little transport vesicles pinching off, moving toward the Golgi. They fuse together until we have our first layer. Then we get processing as these layers move closer and closer to the surface until this top layer disintegrates into all these transport vesicles. And here is a transport vesicle that is destined to be secreted. It comes and it binds to the surface. What would this process, how would we categorize this process? Taking material and spitting it out. What kind of cytosis? Exocytosis. So exocytosis is sort of the very end of this whole process. It started in the ER, modified in the Golgi, and we finished with exocytosis of material exiting the cell. Now, also coming through the ER and the Golgi are these little vesicles called lysosomes. Lysis. You remember when we were talking about molecules that we're putting together or breaking apart? What does lysis mean? Lysis, break, separate. In this case, lysosome have all of these enzymes present inside their little vesicle. So that when we bring in, let's say here's an organelle. It says worn out organelle. Isn't that technical? Well, we've got to process this organelle, so we put some lysosomes into it. Those lysosomes destroy it, break it down, and we undergo exocytosis to get rid of the waste. If there's anything that can be recycled, we recycle it back into the cell. But in this case, it's the lysosome that's going to break that material down into its basic building blocks or the wastes that we get rid of. So, hydrolases, those are a specific kind of enzyme that's going to be great at breaking down materials, particularly proteins, carbs, and fats. We say cellular recycling, that really should be organelle recycling, much that we have illustrated here. Now, I think this kind of goes without saying, but can you imagine how dangerous it would be to make these enzymes and just be free-floating loose inside the cytoplasm of the cell? You would self-digest. So it's important as we're moving into the Golgi that packaging and sorting are putting those lysosomes together, and we're not putting the lysosomes with anything else. If they were all together in the same vesicle, then they would all be destroyed. But the lysosomes are protected in their vesicle, and they only bind with the material that they're supposed to bind with. Again, we just came through this. They are first made in the rough ER. They go through the Golgi complex, and don't worry about this. We don't need that level of information here. This is just an example of going through our endomembrane system to produce these really important little vesicles. All right, with the time we have left, that was a lot. I mean, that was a ton. Endomembrane system. Now I want to get into probably one that you are more familiar with than all of those others. You probably got the nucleus, but ER, Golgi, you probably didn't have so much on that one. The mitochondria functions as a site of respiration. What's another way of saying that? It's the powerhouse of the cell. You can watch that on Saturday morning cartoons. One of my favorite, I have two granddaughters, 4 and 6, and one of my favorite little things to watch with them, other than now Barbie, is if you go on YouTube, there is a group and a channel called Amoeba Sisters. They have some cartoonists. It's kind of like Schoolhouse Rock for biology. They do a really great job of explaining biological processes, and it's accurate. It's really accurate, and it's kind of fun to watch. I actually use a lot of Amoeba Sisters in my science major, Introduction to Cell and Molecular Biology, because some of the details gets really, really technical. So, just like we saw in a nucleus, mitochondria have a double membrane system. The outer membrane, this is going to be adjacent to the cytoplasm. That should be cytoplasm there. The inner membrane is all folded up. You can't really see it there, but you see these lines inside this little capsule-looking thing? These are folds of the inner membrane, and those folds are called cristae. Now, in biological systems, where you see stuff folded up like this, what do you think is the benefit of having all those folds? Your brain, for instance, has all these folds in it. Your intestines have all these ridges and folds in it. What's the advantage of the folds? You can fit more. That's a different way to say you've increased the surface area. And so what you're fitting on more of these folds inside the mitochondria are the proteins responsible for producing energy, for producing ATP. If they're membrane proteins, certainly the more membranes you have, the more of them you got, the more energy you can make. Now, do you remember with our nucleus and that double membrane, we said evolutionarily one cell got with another and that brought that membrane with it? Well, there's a theory that mitochondria were like bacteria, and that these bacteria were good at making energy, and this one cell that had just picked up all this genetic material and said, hey, you can make a lot of energy. Why don't you come live with us? I've got this nucleus now that can organize all of this information. I've got a lot of raw material, but you make a lot of energy. Come in here. Let's set up a partnership. And that's how it's thought to have picked up the outer membrane of the mitochondria. And another piece of evidence that would suggest that evolutionary possibility, bacteria have circular chromosomes. Guess what your mitochondria has? It's got its own DNA. And it's a circular piece of DNA, and many of those genes fit with and match with what we see in bacteria. Mitochondria actually undergo their own kind of division just like bacteria. It's really kind of freaky. But the cool thing about this, though, when you look at mitochondrial DNA, scientists, this is called a cladogram. I'm not going to test you on what that is and everything. But scientists looked at the mutation rates and the sequences of mitochondrial DNA. Because unlike your mom and your dad's DNA that kind of got mixed up to make you, because you probably look maybe a little bit like one, a little bit like the other, but you don't look exactly like them, your mitochondrial DNA is exactly the same, unless there was a mutation, as your mother's mitochondrial DNA. Because what do you get from dad? What do you get from dad? Don't say money. What do you get from dad? You get half a set of chromosomes. That's it. That's all you get from dad. What do you get from mom? Everything else. You get all the membranes, you get all the Golgi, you get all the ribosome. That's like dad being lazy, right? But you got all the mitochondria that you have from mom, so your mitochondrial DNA is identical to your mother's. So scientists looked at the mutation rates and the sequences of mitochondrial DNA of humans across the planet. And when they compared them all together, they could trace back to what the mitochondrial DNA of the original Eve, this was maybe 20 years ago on a National Geographic's cover. And when they look at the sequences of mitochondrial DNA, guess where the first human came from? Southern Africa. And so we get our mitochondrial DNA, and when you look at the differences and the predicted migration and the change, here we've got the African group. It comes up here in the European group, moves over into the Asian group, Australian group. We come up over here into the Russian group, across the Bering Sea, North America. South America is the most recent changing that we see. That follows with our anthropological data. It follows with the fossil record. So your mitochondrial DNA supports everything we see about human evolution and the migration of humans out of Southern Africa.